All right, good morning. So another day, another polymer, biopolymer filament. Today we have microtubules, which will be our final filament system that we'll discuss, and it'll take us through some interesting new directions and new types of biology. So microtubules. So first thing we want to talk about with microtubules is their function. What are they used for in cells? And I have a few different sort of examples of some of the biological functions of microtubules that illustrate the specific cellular roles that microtubules play. So in proliferating cells, microtubules are the key structural elements in mitosis, in the mitotic spindle. And in the mitotic spindle, the microtubules give the spindle structure, and they're responsible for generating forces. So structure and forces. And it's because of the role of microtubules in the mitotic spindle, which is something that forms only in proliferating cells, that microtubules have been targeted for cancer chemotherapy, okay, because the mitotic spindle is something that's only formed when cells divide and grow. Another role for microtubules in mammalian cells, where microtubules are absolutely critical, is in fertilization and fertility. So in the oviduct, the microtubules are a key component of the cilia that move the ovum along the oviduct. And for sperm, the microtubules are the main components of the flagella. So in this sense, then, the microtubules are key elements of motile structures. Okay, that underlie various forms of cellular motility. In most cells, the intracellular organelles have a very specific kind of organization. You've seen the Golgi and the endoplasmic reticulum, and microtubules play a crucial role in the internal organization of cells. So in this sense, the microtubules act as scaffolds for many components of the cells, such as the Golgi apparatus, the endoplasmic reticulum, and many other cellular structures. And when the microtubules are disrupted, which happens when cells enter mitosis, these organelles become disrupted too. Okay? So we can also illustrate some of the functions of microtubules in cell shape and in intracellular motility by looking at a highly specialized cell, which is a nerve cell. So nerve cells have an axon, and they have dendrites. And microtubules are central for developing this elongated shape of the neuron, okay? They resist compressive forces. They're much better at doing this than actin filaments. Actin filaments have to team up, okay? They're kind of wimpy compared to microtubules. Microtubules are big and strong, and they're really good at resisting compressive forces. this axon is completely dependent on microtubules for its structure. If you depolymerize the microtubules, this whole process will collapse into the cell body. So if you were to look at a cross-section through an axon, what you would see is that it is chock full of microtubules. In fact, you would see a second filament, which we talked about last time. Remember what those are? These processes. What else do we find in these axons? What did we talk about last time? We had a quiz. You guys were like, okay, in shock from the quiz maybe. Pardon me? Intermediate filaments. And which kind of intermediate filament? Neurofilaments. Okay, so if we look in cross sections of an axon, what we'll see is that they're chock full of microtubules and then neurofilaments. Okay, so the microtubules then are play an important role in determining cell shape, but they play a second role in cells, and that in cells like neurons, that's just as crucial as their role in shape, and that's a role in intracellular motility. So it turns out that most of the biosynthetic material for this neuron is in its cell body, okay? But it's continuously making stuff like neurotransmitter that's getting used really far away from the cell body. You know, the longest axon in your body is like 1 meter long, okay? That goes the length of your leg, from your spine to your leg. And so what has to happen is stuff that gets synthesized in the cell body has to get transported along this axon. And in fact, stuff also is getting transported from the synapse back to the cell body. And that happens along microtubules. The microtubules are highways that are very important for transport of components in both directions. And we'll be learning Wednesday about the motor proteins that mediate this transport along microtubules. That transport is very pronounced in a neuron, which has this very exaggerated kind of morphology, but it occurs in all eukaryotic cells. Okay? So, cell shape, intracellular transport. Okay. So that gives you sort of an overview of some of the things that microtubules do. Now we want to talk about how it is that microtubules do all these things. And so first, we have to look at the structure of microtubules and then at their assembly properties. Again, just as with actin, we had to learn about how actin assembly is regulated to understand how actin functions. We have to learn how microtubules assemble. So let's talk about the structure of microtubules. Function, structure. Okay, so first of all, the subunit of a microtubule is a heterodimer that consists of one alpha and one beta subunit. Okay? Both of these subunits are about 55,000 Daltons or kilodaltons in molecular mass. Okay? And they're related to each other. Alpha and beta tubulin are about 30 to 40% identical to each other. Okay, hence they're both called tubulins, alpha and beta tubulin. Okay? So the other really crucial property of alpha and beta tubulin is that each binds to GTP. Okay, so with actin, actin subunits bind to ATP, but tubulin binds to GTP. So it turns out that alpha binds to GTP, and it does not hydrolyze or exchange that GTP. But beta binds to GTP, and the GTP gets hydrolyzed, and then an exchange reaction can occur to replace that hydrolyzed GTP with GDP. Okay? So, now, we've talked in this class about F-actin. F-actin filaments are about 7 to 8 nanometers. Okay. We talked about intermediate filaments, and they're about 10 nanometers. Microtubules are sort of the king of the filaments because they are 24 nanometers. Okay? In diameter. These are diameters. This is about the same dimensions as a ribosome, okay, just to give you some sense of scale. They're really thick filaments. Does anyone know how thick the lipid bilayers are, like in the plasma membrane? 7? 7? Wait, what units are you using here? 70? Did you say 70? 70 what? Angstroms? Is that what you said? No, that's right. Well, I don't know if you said it, but so but let's keep it in nanometers. So they're sort of in the 5 to 6 or 7. I usually use 5 to 6 nanometer for bilayers. But of course, it varies with the specific lipids that are involved. So the bilayer is actually really thin compared to the thickness of these filaments. I mean, a cell is really like a bubble, a soap bubble or something. The lipids are, a lot of people are really surprised when you think about the scale of these things, that the lipids are really very thin. They have this really important property that they're hydrophobic and they don't let water or they don't let hydrophilic molecules or charged molecules pass through them, but they're very thin. Okay. Now, the other important feature of microtubule structure is how the subunits are arranged within the microtubule. Okay? So microtubules are made up of protofilaments, which are single chains of tubulins. These are now alpha-beta tubulins. I'm considering an alpha-beta heterodimer as one subunit, okay, to save myself from drawing alpha-beta, alpha-beta. But these are arranged in protofilaments, and then those protofilaments are joined together to make a tube. And typically, one of these tubes is composed of 13 protofilaments. Okay. However, there's some plasticity in the microtubule, and the number of protofilaments that can be found in a microtubule can actually vary in nature from 11 to 16 protofilaments. Okay. So the other important aspect of this, filling out this structure, is that like actin filaments, there's polarity in the microtubule. And I'm talking about a structural polarity, as I was talking about with actin filaments. So they have a minus end and a plus end. The alpha betas are lined up in sort of a polar manner. So the alpha subunits, if you were to look at the microtubule from the minus end, you would see just alpha subunits. And if you were to look from the plus end, you would see beta subunits. Okay? So the filament has polarity, like an actin filament, and that turns out to be very important, the polarity, for two reasons that's important. It's crucial for thinking about the dynamics and for thinking about how motor proteins interact with the filament. Because what we'll see on Wednesday's lecture is that there are a variety of motor proteins, kinesins and dyneins, that see this lattice differently. Okay? A dynein sees this lattice and wants to move towards the minus end, and most of the kinesins want to move towards the plus end. Okay? And that's the basis for this kind of trafficking that one sees along neuronal processes. Okay, so let's talk then about the assembly of microtubules. Okay, so first of all, so that like with actin, the dynamics and the assembly properties are crucial to the function of microtubules. How do we work with tubulin in a test tube? So if we want to study microtubule assembly, at 4 degrees, the microtubules are disassembled and we get monomers. Okay. But if you warm the solution up to 37 degrees, you'll get microtubules. Okay. So that actually makes the basis for methods used to purify tubulin. What one does is to create a brain extract. Why brain? Why would you think we use brain to make microtubules, to make tubulin? Pardon? Neurons. Yeah, what about neurons? They're rich. Yeah, neurons are really rich in tubulin. So a brain, 20% of the protein in your brain is tubulin, okay? You don't have to be a very good biochemist to purify something when the theoretical yield is only 5-fold, okay? So there's tons of tubulin. Biochemists like to look for sources that are where their protein is very abundant. So in tubulin, if 1/5 of your protein is tubulin, you just need to purify it 5-fold to have pure tubulin. And so what you do is you make an extract from brain in the cold, and then you warm it up to 37 degrees, and the microtubules polymerize, and so you spin the extract, and the microtubules are collected out. You can then take those microtubules and chill them, and they'll fall apart to tubulin again. Okay? So it's actually a pretty easy protein to work with. So let's look at the assembly kinetics. So they look a lot like those for actin. If we look at assembly versus time, we have a lag phase, a growth phase, and then steady state. Okay, we've seen that before. If we add seeds, we can eliminate the lag phase. And assemble microtubules right from the start. So what that tells us is that, what does that tell us? Pardon me? It's similar to actin in what sense? Yeah, so it's similar in that it has a lag and elongation phase. So specifically the lag, what does that tell us about making a microtubule? Yeah. An unstable intermediate, right. So it's hard to start. It's hard to get it started, and once it starts, it gets going very robustly. And so the reason that it's hard to get a microtubule started is because the structure is complex, okay? So we have to make 13 the protofilaments, and the protofilaments then have to associate with each other in order to grow a microtubule, okay? So there's a lag phase. Okay. Also like actin, and even more important for microtubules, is there's a delay between assembly and nucleotide hydrolysis. So as a microtubule grows, this is its growing end, and the plus end is the fast-growing end, the subunits that associate are GTP subunits, and then there's a lag, and as you go deeper into the microtubule, the subunits are GDP subunits. Okay? So what people say is that there is a GTP cap at the plus end. And we'll return to this in a few moments. That becomes extremely significant for understanding microtubules. So, okay. So getting these microtubules started is really hard. So nucleation then becomes key. And this provides, as with actin, it provides an opportunity for regulation. So in cells, nucleation of microtubules is regulated like it is with actin filaments. And so it turns out that in cells, there's a structure that nucleates microtubules, and it does this in a manner so that the plus ends grow outward. Okay, so if you look at microtubule organization in a cell, what you almost always see is next to the nucleus, there's a structure called the Microtubule Organizing Center, or MTOC. Okay. This is also referred to as a centrosome. Essentially, all of the microtubules in the cell emanate at this centrosome, or MTOC, and they project out into the cytoplasm in a radial manner such that their plus ends are free in the cytoplasm, and their minus ends are buried in this MTOC. Okay? So now I'm going to tell you something that simplifies your life a bit in thinking about microtubules and microtubule dynamics. The minus ends are essentially never free inside cells, only the plus ends. So when we think about microtubule dynamics, we'll only have to think about their plus ends and what's happening at their plus ends. Okay? So this MTOC nucleates the assembly of microtubules. How do we know that? We can depolymerize all the microtubules in the cell by using a drug or cooling the cells down to 4 degrees. The microtubules will depolymerize, and then we can change the conditions so assembly is favorable again. And what we see is that the microtubules all grow out from this microtubule organizing center. So what's in that microtubule organizing center that's so important for their ability to nucleate microtubules? Well, they have at their center something called gamma tubulin. Okay, so gamma tubulin is related to alpha and beta tubulin. It's a third member of the tubulin superfamily. And gamma tubulin forms into structures that are sort of like lock washers. They're like that, if you know what a lock washer is. And these structures contain gamma tubulin plus several other proteins. And these are what nucleate microtubules. And so they're kind of analogous to the ARP2-3 complex, where in that case, there are two proteins that are related to actin, ARP2 and ARP3, that nucleate the assembly of a filament. Here we have gamma tubulin in this ring structure, which is referred to as gamma Turk. This stands for gamma tubulin ring complex. Okay? And so this ring of gamma tubulin acts to nucleate the assembly of microtubules. Okay? And it turns out that gamma Turk complexes are embedded, so these complexes are embedded in the centrosome, or MTOC. And so if you look at these centrosomes, what you see in by electron microscopy is actually a lot of fuzzy material. And then at the center, you see a pair of structures at right angles to each other, which are the centrioles. Now, centrioles are made up of a number of proteins, but the major proteins are tubulin. And they have an interesting structure. If you look in cross-section in the EM, what you see is that the centrioles are made out of 9 microtubule triplets. And so these are arranged in a manner like this, where there's one complete microtubule and then 2 partial microtubules. And they radiate out like this, and there's 9 of them around a circle, and there are better pictures in your handout for today's lecture. Okay, so these are rather elaborate structures, but it turns out that they have no role in nucleating microtubule assembly. Okay, what is their role? Well, that's been a subject of some interest and debate. One of the things that they do is that they can become basal bodies which are the templates for the formation of cilia and flagella. But the bulk assembly of microtubules is actually due to nucleation of the microtubules from this fuzzy material. And what happens is the gamma-Turk complexes are embedded in this fuzzy material, and that's actually what nucleates microtubule assembly. These centrosomes are kind of have an interesting place in the history of cell biology because people people have been able to see structures inside cells for a really long time. And if you look at some of the papers that were being published like 100 years ago, where people would look at, say, a cell that's migrating, what they would see is that the centrosome was always leading the way, okay? And so people talk about the centrosome. So if this cell is migrating in a direction like that, the centrosome always leads the way. And so people thought that, well, maybe the centrosome has something to do with the guidance system for the cells. And another thing that I find kind of amusing in these old papers, if you see the mitotic spindle, so when a mitotic spindle forms, the centrosome divides, okay? Each centriole duplicates, it divides into 2, and then you get 2 of these asters of microtubules that I've hidden now, and you get a mitotic spindle, okay? And so then the cell's going to divide, and we all know now that the spindle is what divides our DNA at mitosis. But if you read these early papers, they speculate that the mitotic spindle was a structure whose main objective was to faithfully segregate a centrosome to each daughter cell. Okay, so they thought that the important cargo and the important activity of the spindle was to separate centrosomes. They didn't know about the DNA at that point. That came a lot later. So centrosomes have kind of interesting behaviors, and they've fascinated cell biologists for a really long time. Okay, so let's talk now then about the dynamic behavior of microtubules. So I told you that when microtubules assemble, there's a delay between the association of a new subunit with the plus end of a microtubule and the hydrolysis of the GTP. And so what you get are microtubules in which you have GTP caps, okay, and then GDP in the body of the microtubule. So when GTP is at the plus end remember we only have to think about plus ends, is at the plus end of the microtubule, that microtubule is stable. And not only is it stable, but it will continue to add subunits and it grows. Okay? Now occasionally, there's some kind of stochastic event where hydrolysis catches up to the end of a microtubule. And when that happens, you end up with GDP all the way through the microtubule. Okay? That microtubule is unstable, and it shrinks rapidly. Okay? So it shrinks or disassembles. And so basically what you have is a two-state system in which microtubules can grow or they can shrink. And what one sees when one observes microtubules is you see even at steady state, when there's no net increase or decrease in polymer, that all microtubules they're not treadmilling where they're keeping a constant length but adding subunits at one end and losing subunits at the other. Every microtubule in the population at steady state is either growing or shrinking. Its length is changing continuously, and it transitions between these two states. A given microtubule can be growing and stop growing and shrink back, then stop shrinking and start growing again. This behavior, which is the most crucial thing that you need to understand for this part of the class, has a name, and it's called dynamic instability. And so if we were to look at the plus end of a single microtubule, next time I'll bring some movies to show you of this behavior. So one of the nice things about microtubules is that because they're so big and because of their organization, how they're more splayed out in cells, both in a test tube and in living cells, you can actually watch the dynamics of single microtubules. And that's something that you cannot do for actin filaments. Actin filaments are smaller and they're much more densely packed together. And so although we have a lot of theory about how they should assemble and disassemble, we can never really observe the individual filaments. But in microtubules, there's absolutely no ambiguity to how they behave because we can observe them and make videos of them. So if you were to look at a single microtubule and to look at its length as a function of time, and let's just say this is 50 micrometers, and we'll look at it for 60 minutes. Just put some numbers in here. What you would see is that the microtubule would be growing, and then it would shrink, and it might grow some more, then it might shrink a long way, then reverse and grow, shrink, and grow again. So a given end of a microtubule is constantly going through these conversions, and it's constantly dynamic, okay? It's never static. So when there's some more terminology here, when a microtubule is growing and then it suddenly starts shrinking, that's called a catastrophe. And when a microtubule is shrinking and then starts growing again, here, then that's called rescue. Okay? And so microtubules convert between growing phase and a disassembling phase. And this is the behavior that one sees for microtubules in a test tube at steady state and in a living cell. So obviously that takes a lot of energy to constantly be adding tubulin subunits, converting the GTP to GDP. Why would a cell do that? Does anyone have any idea? What's the value of having microtubules constantly growing and shrinking? Cell movement, okay. So, but what specifically do you think is the value of having microtubules that continuously grow and shrink. Wouldn't you want to just, build some microtubules, decide what structure you want to build in the cell and just build it? Why build things that keep falling apart? It seems like really futile, right? The cell itself is not static. That's a really good thought. And so in what way is it not static? Okay. It's transporting materials across microtubules. But if you were transporting materials, say, along one of these neural processes, wouldn't you want just to have a stable highway there that you could transport stuff along? Is that, another idea in the back? These are all good ideas. So. More efficient? Efficient for what? Well, that's an interesting idea. I like that. That's an interesting idea. Okay. That's a good, I think I agree with that, yeah. So to perform useful work like separating chromosomes, yes, I would agree with that too, that you could harness this mechanism, and we'll talk about mitosis and in fact how this works. Yeah, one more and then yeah.
Does it have to do anything with the reserve also of GDP versus GDP?
So does it have to do with the reserve of GDP versus GTP? Well, the reserve in cells, GTP levels are much, much higher than GDP, sorry. So you're going to have a big reserve of GTP tubulin, and that tubulin is there to assemble, okay? So let's paint a picture of what's going on here in the cell. So you guys have a lot of good ideas and thoughts, and let's see. So here we have our MTOC, and what I'm telling you is that in the cell, this MTOC is nucleating microtubules, and that some of these microtubules at any given time are growing, and at any given time, some of them are shrinking. Okay? And then what's the use of that? Well, the use of that system and that design is that it is a very adaptable and responsive and evolvable system. Okay? And so what that means is that if this is a cell, okay, here we have sort of a generic cell where we have the MTOC and the nucleus. We know that microtubules, okay, there's only one real kind of tubulin in cells, and the alpha-beta dimer, and they make microtubules, and we know that those microtubules, though, can form all sorts of structures. So the microtubules can form a spindle. They can form a neural process, where I'll draw sort of a bigger view of that neuron over there, where you have all these microtubules in one direction like that. Okay? And so how do you build a system where you can start with sort of a generic cell like this, and then rearrange the microtubules in response to stimuli from the environment? One cue might tell the cell to proliferate, to divide, to enter mitosis. Another cue from the environment might tell the cell, grow a process in this direction. So how do you take a building block, a structure, the microtubules, and how do you build into it an adaptability and responsiveness that allows those structures to adopt many different kinds of organizations, and to do it rapidly, and to do it efficiently, and to do it reversibly? Well, so here we have a system where we have a microtubule organizing center. And what does it do? It's generating microtubules that are continuously sprouting out from that microtubule organizing center. Those microtubules, you can think of, are sort of probes. They're probing the three-dimensional space of the cell. microtubules will grow out, grow out, grow out, grow out, grow out in all different directions, and usually they'll shrink back. Okay? But suppose we had a signal in the environment that impinged upon the cell. And that signal caused a change at the surface of the cell in one section of the cell, so that when a microtubule grew out in this part of the cell, that microtubule became stable. Okay? When its end touched this part of the cell, it became stable. Okay? Imagine that you made a movie now of this cell over time. The microtubule organizing center is going to continue to spew out these microtubules in 3 dimensions, but any microtubule that happens to come into this part of the cell is stabilized. Okay? So very quickly, what you can do with such a system is evolve a new organization. You can rapidly reorganize the whole context and shape of the microtubule cytoskeleton by a cue at the surface of the cell that changes the probability that that microtubule will disassemble. If it reaches that part of the cell, it's unlikely to disassemble. Likewise, one second, if you have a cell that has chromosomal DNA in the middle, and you've duplicated your centrosome, and you give the chromosomes properties such that any microtubule that comes in contact with this DNA becomes stable. So you have sources of microtubules, again, that are projecting microtubules in 3 dimensions and probing all the surfaces of the cell, but you make it such that when a microtubule contacts the DNA, it becomes stabilized. You very quickly evolve a mitotic spindle. Okay? So the purpose of dynamic instability and the beauty of this system is that it allows this network of proteins, the microtubule network, to change its organization in response to cues and to develop new structures for new functions. Okay? And if you want to change the organization at a later time, all you have to do is turn off that signal that stabilizes the microtubules in that region of the cell. Okay, question here.
So these new tubules are forming at any given time randomly, and then if they don't get a signal that stabilizes them, they just degrade?
That's right.
Like, there's no regulation of nucleation of the tubes?
There's no regulation of nucleation of the tubes. The only regulation, which we'll return to when we talk about mitosis, is that as a cell enters mitosis, there's a general upregulation of the activity of the microtubule organizing center, so it's making more microtubules, okay? Because these microtubules have to very quickly probe space in order to build a mitotic spindle. Okay? Another question. No, that would be that would be an opportunity for the cell to regulate if there was a mechanism that turned off the shrinkage. But it turns out that's a stochastic process, okay, that it just happens randomly. Sometimes the microtubules actually shrink all the way back to the MTOC, but other times they just stop, okay? What causes them to stop? Well, so what's going on at the end of these microtubules? So as the microtubule grows, it turns out it has a different structure from a microtubule that's shrinking. And there are pictures in your handout, but this, a growing microtubule has sort of a smooth surface. Sometimes the protofilaments in one part will be extending beyond the other part as it's growing. This is from EM studies of growing populations of microtubules and shrinking populations of microtubules. When the microtubule shrinks, the protofilaments begin to peel back just like a banana peel, okay? Why does this happen? Because when GTP is hydrolyzed in the microtubule, this puts a strain on the microtubule lattice the GTP subunits in this microtubule are happy, okay, to be there. But when GTP is hydrolyzed and the PI dissociates, now that subunit is locked in a structure that it doesn't want to be in. It's under strain, okay? And the way we know it's under strain is because once it starts to depolymerize, the protofilaments start curling backwards, okay? And so it's thought that the structure is part of the key to these transitions, okay? So this structure is stable and likes to grow, and this structure is unstable and likes to stop growing. And so what happens is sometimes these protofilaments all depolymerize off the end, and the end looks like a growing end again, and then it can start to grow again, okay? So it's kind of hard to transition from one structure to the other structure, and once you're in that structure, you'll continue to do whatever is favored by that structure for a while. And that's why the microtubules have this persistence in their behavior, where they'll continue to grow or shrink for quite a while before they do these transitions, okay? So in a way, this is almost sort of like a Darwinian concept, where the MTOC is generating variation by probing different kinds of possibilities, and then there's a selection for a certain subset of the possibilities, and that becomes, then the cell sort of evolves that kind of an organization. Yeah, another question.
So what does the signal do to stabilize the, does it like stabilize the cap?
So it could work by either stabilizing the GTP cap or by just stabilizing the subunits, like cementing them into the filament. How would this such a stabilizer work? And the ones that I know about work in the latter mechanism. They're sort of like, for actin, we talked about tropomyosin or phalloidin, stabilizing the actin filament. Similarly, there are proteins that will stabilize the lattice. Okay? Okay. So, I'm sorry, I should have mentioned that. So, microtubules and actin filaments can only assemble or disassemble from their ends. And for microtubules in cells, we only see assembly and disassembly happening at the plus end because the minus end is buried in the microtubule organizing center. So I wanted to mention two tools we use to study microtubules. There are a variety of drugs that bind to microtubules and tubulin, just like there were for actin. So Taxol comes from the Pacific yew tree. It's a natural product. And because Taxol blocks mitosis, Taxol has been used for cancer chemotherapy, and it's actually one of the most effective drugs for ovarian cancer and breast cancer therapy. And what Taxol does is Taxol stabilizes microtubules. Okay? So you could think Taxol is to microtubules as what is to actin filaments? Phalloidin, good, okay. So another drug is colchicine, which is another natural product that comes from a plant called the autumn crocus. That's actually not that important, but what cytoclasin does is a little bit different from the actin drugs because it binds to both the alpha-beta and it binds to the ends of microtubules. So when it binds to the ends of microtubules, it inhibits the further assembly, okay? And therefore, the filaments have an opportunity to transition to a GDP state, and therefore they tend to disassemble, okay? So it can bind to the alpha-beta subunits, but it can also poison the ends of microtubules and stimulate the disassembly of the microtubules just by binding to the ends and preventing further associations of subunits. So it's a little bit like cytoclasin was for actin. Okay, any more questions? Okay, that's it for today then.